Cell membrane structure and function. The plasma membrane plays a key role in the behavior of cells, which includes cell-cell communication, adherence to other cells or structures, cell movement and migration, access to nutrients in the cell microenvironment, and immune recognition. Plasma membrane alterations in malignant cells can be inferred from the various properties associated with their growth and behavior, such as lack of density-dependent growth inhibition, loss of anchorage dependence, decreased adhesiveness, and invasiveness through normal tissue barriers. In addition, several biochemical changes on malignant cell surfaces have been observed, i.e., the appearance of new surface antigens, glycolipids, proteoglycans, and mucins, as well as altered cell and cell ECM, extracellular matrix communication. Modification of extracellular matrix components. The ECM is involved significantly in regulation of cell proliferation and differentiation. It is now known that interaction with mesenchymal stroma on which tumors grow and development of blood supply plays an important role in growth, invasion, and metastatic potential of tumors. Continuous remodeling of the supporting stromal structure takes place by interaction of the grown tumor with host mesenchymal cells and vasculature. Tumor cells can breach the basement membrane, a specialized type of ECM barrier by release of a variety of proteases, collagenases, and glycosidases that can degrade various matrix components. This allows invasion of tumor cells across tissue barriers, blood vessels, and lymph channel walls. Additionally, certain matrix components can be secreted by the malignant cells and possess receptors for certain components. This capacity is known to enable the binding of malignant cells to the vascular endothelium and is suggested to be involved in tumor metastasis. The host mesenchymal cell and proteoglycan types can be modulated by polypeptide factors released. Mediums such as agrose or methylcellulose without attachment to a surface. This property is termed anchorage-independent growth. This property has provided a way to compare the growth properties of malignant and normal cells and also is a technique used to test the sensitivity of cancer cells to chemotherapeutic agents and for screening potential anti-cancer drugs. Loss of cell cycle control and resistance to apoptosis. When a variety of suboptimal growth conditions are presented, normal cells respond by entering into the quiescent phase, G0 phase, of the cell division cycle. A decision regarding commitment to continue into the S phase is made in the G1 phase. Cells decide whether to stop in G1 or wait until optimal conditions return such that cell division can occur. Once cells become committed for division, they continue through S, G2, M and then return to G1. If cells are blocked during cell division, i.e., in S, G2, or M phase for any length of time, the cells die. This control is lost in the cancer cells. For example, accumulation of DNA damage in normal cells due to X-ray radiation or ultraviolet UV causes G1 arrest, allowing repair of damaged DNA prior to DNA replication. Similarly, the G2 phase checkpoint allows repair of damaged chromosomes before they are segregated in mitosis. In either case, irreparable damages lead to cell death. Cancer cells possess compromised or absent checkpoint controls, and hence continue through the cell cycle in spite of the accrued DNA damage, leading to persisting and accumulating mutations. Interestingly, the loss of cell cycle checkpoint control in cancer cells may also attribute increased susceptibility to anti-cancer drugs. Normal cells possess protection mechanisms against exposure to toxic agents or growth-limiting conditions, i.e., checkpoint control mechanisms are activated. In contrast, cancer cells escape these checkpoint controls and continue into cell cycle phases, which increases their susceptibility to cytotoxic effects induced by irradiation or drugs. By tumor cells. Also, factors favoring proliferation and invasiveness of tumor cells can be released by tumor stromal cells. Activated fibroblasts in the tumor stroma release several growth factors that stimulate cell proliferation, inhibit apoptosis, alter cell differentiation, and that upregulate proteases involved in degradation of ECM. These factors include hepatocyte growth factor HGF, insulin-like growth factors IGF-1 and -2, EGF, TGF-A, TGF-B, interleukin 6, fibroblast growth factors FGF-2 and 10, and matrix METALLOPROTEASES1 and -7. A number of potential targets for anti-cancer therapy are provided by the multiple effects of the tumor stroma on cancer growth and progression. Cell extracellular matrix and cell adhesion. Cells in tissues are attached to the ECM and to one another. When these adhesion events are disrupted, increased cell motility and also an increased potential for invasiveness through the ECM is acquired. For most cell types, it is necessary that they are attached to the ECM to maintain their normal function, growth, and differentiation. It is this attachment that attributes the property of anchorage dependence to the cells. If normal cells become detached from their ECM binding, they undergo apoptosis. On the contrary, less dependence on this attachment is observed in the tumor cells, and they are free to proliferate, invade tissues, and wander. Cell surface receptors called integrins mediate the adhesion of the cell to the ECM. Integrins enhance mitogenic signaling by cooperating with growth factors. In human breast and colon cancer tissue and in chemically transformed human cells, alterations in integrin receptor expression have been observed. For normal cell proliferation and differentiation, cell-cell interactions are also important. A family of molecules called CAMs, cell adhesion molecules, mediate these interactions. CAMs function as both receptors and ligands, i.e., they are receptors on one cell and also act as a ligand for another cell. CAMs include a large family of molecules, and cadherins superfamily of CA2+ dependent transmembrane glycoproteins are one among them. Cadherins play a key role in cell-cell contact initiation and stabilization. E-cadherin is expressed in epithelial tissue and is the predominant type of cadherin. Altered expression and function of E-cadherin have been reported in human cancers. Additionally, increased invasiveness, metastasis, and poor prognosis have a correlation with E-cadherin downregulation in cancer patients. By transfecting E-cadherin cDNA into carcinoma cells, this invasive phenotype can be suppressed. Also, E-cadherin gene transfected cells can be exposed to E-cadherin antisense RNA or E-cadherin antibodies, and the invasiveness property can be restored. Germline E-cadherin gene CDH1 mutations have been found in New Zealand Maori families, and these individuals show susceptibility to gastric cancer that is dominantly inherited. Beta-catenin is the cell surface receptor for E-cadherin, and early mutations in the progression pathway of human colon cancer affect cellular beta-catenin distribution. The development of cancer. Early studies of virus-induced tumors gave emergence to the idea that cancer development is a multi-stage process. Peyton Roos and his associates, who worked on virus-induced tumors, proposed that induction and successive growth of cancers involved different mechanisms, and called them as initiation and promotion, respectively. Discovery of chemicals that could initiate or promote tumor formation contributed toward understanding the process of cancer development. Two groups of chemical agents were discovered: those that could initiate a tumor, e.g., benzo, pyrene, urethane, etc., called carcinogens, and those that could promote cancers, e.g., creosote oil, croton oil, etc., called as cocarcinogens. The chemical agents that could do both were called complete carcinogens, e.g., polycyclic aromatic hydrocarbons, certain aromatic amines, aflatoxin B1, and nitrosamines. Hence, studies involving virus-induced and chemical-induced cancers indicated that carcinogenesis is a multi-stage process. Both initiation and promotion phases of carcinogenesis were studied using chemical agents that were primarily initiating, e.g., urethane, or those with only promoting activity, e.g., components of croton oil. Even low doses of complete carcinogens were considered for studying initiation. Mouse skin carcinogenesis was used as a model to study the initiation-promotion phases of cancer. Single application of an initiator, urethane, typically brings about tumor initiation. Alternatively, low-dose subcarcinogenic dose of an initiating and promoting agent, complete carcinogens such as benzo, a pyrene, a polycyclic hydrocarbon, can be applied for initiation. Promotion is carried out by applying a formyl ester, such as TPA, repeatedly, e.g., 3 times a week. At 12 to 20 weeks, benign papillomas begin to appear, and by about 1 year, squamous cell carcinoma develops in 40% to 60% of the animals. Usually, no malignant tumor is formed if only the promoting agent is given, or if the promoting agent is given before the initiating agent. Carcinogenic progression is an extension of the tumor promotion stage, i.e., cell proliferation caused by promoting agents allows propagation of cellular damage inflicted by initiation, and causes clonal expansion of the initiated cells. When damaged cells consisting of genetic alterations are propagated, it leads to the acquisition of more genetic alterations, i.e., these cells exhibit genetic instability, which is a hallmark of the progression phase. This leads to chromosomal aberrations such as translocations and aneuploidy frequently observed in cancer cells. Such genomic alterations of the neoplastic cell in the progression phase increase the growth rate, invasiveness, and metastatic ability of advanced neoplasms. Multistage development of malignant tumors has also been observed in tumors of the thyroid, lung, mammary gland, urinary bladder, and in cell culture systems. Therefore, this multistage development is a general phenomenon. Experimental evidences are consistent with a clinical history of human tumor development after exposure to known carcinogens. This involves an initial exposure to a known physical or chemical carcinogen, a long lag period during which a probable exposure to promoting agents occurs, and finally, the development of a malignant tumor. An insight into the mechanisms involved in these processes has been obtained by various characteristics of tumor initiation, promotion, and progression. Tumor can be initiated after a single, short-term exposure to a potent initiating agent. In the mouse skin system, actual initiation events that lead to the transformation and formation of a dormant tumor cell seem to occur within one meiotic cycle, i.e., about 1 day. Additionally, the initiation seems to be irreversible. The exposure to a promoting agent can happen even a year after initiation, yet a high percentage of tumor development will still be observed. Thus, only a small amount of time is required for the initiation phase, and it is irreversible. The malignant alteration that an initiated cell undergoes must be heritable as it is conveyed to its daughter cells. These properties corroborate the idea that initiation involves a genetic mutation. On the contrary, the tumor promotion phase is a slow and gradual process requiring prolonged exposure to the promoting agent. Greater part of the latent period of tumor development is occupied by promotion. It is partially reversible, and by using certain anticarcinogenic agents it can be arrested. Tumor promotion consists of a phase of cell proliferation, which allows propagation of the initiated damage, and an altered clone emerges. The promoting agent's function is mitogens for the tissue in which promotion occurs. Progression of a tumor involves continuous clonal proliferation of altered cells. During this process, growth control is lost, and they also escape the host defense mechanisms. These become the predominant phenotypic traits of the proliferating cells and enable progression into a clinically detectable tumor. Due to the marked changes that occur in the genome to lead through tumor progression, it is also thought to be an irreversible process. Mechanisms of tumor initiation. Malignant transformation of normal cells is initiated by a carcinogenic agent and involves a permanent, heritable alteration in gene expression of the transformed cell. This can happen either via direct mutational or genotoxic events, wherein a carcinogen reacts with DNA directly, or by indirect or epigenetic events that alter gene expression without reacting directly with the DNA-based sequence. The mutational theory of carcinogenesis is favored by most researchers, i.e., direct action on the genome is involved in the initiating events. 3 kinds of evidence support the mutational theory. 1. Agents that damage DNA are frequently carcinogenic. Chemical carcinogens are usually activated to form electrophilic agents that form specific reaction products with DNA. The extent of formation of some of these reaction products, for example, alkyl 6-guanine, a DNA duct formed by alkylating agents, has been shown to correlate with the mutagenicity and carcinogenicity of certain chemical agents. Ultraviolet and ionizing radiation also interact with DNA at doses that are carcinogenic. 2. Most carcinogenic agents are mutagenic. Several in vitro testing systems, e.g., Ames tests based on mutational events in microorganisms, have been developed such that a rapid screening of various chemical agents for their mutagenic potential is possible. These tests have shown that the degree of mutagenicity of a chemical correlates with its degree of carcinogenicity in vivo. 3. Patients with DNA repair deficiencies show an increase in the incidence of cancer. Individuals suffering from certain recessive inherited disorders show significantly higher prevalence of cancer than observed in the general population. A common factor in these disorders is a lack of DNA repairability. These individuals lack the ability to repair certain chemical or physical damage to DNA. Higher cancer incidence in these diseases provides the best evidence for the interrelationship between mutagenicity and carcinogenicity. Xeroderma pigmentosum (XP), an autosomal recessive genetic disorder, is one such example. It is the most widely studied disease of DNA repair deficiency, and extreme sensitivity of the skin to sunlight is its characteristic feature. Some form of skin cancer eventually develops nearly in all affected individuals. Additionally, heterozygotes carrying XP gene mutation do not express the disease also seem to develop non-melanoma skin cancers at a higher rate. XP individuals are deficient in the repair of DNA damage caused by ultraviolet, and most of them have a defective excision repair pathway. XP cells also exhibit a lower efficiency in repairing chemically induced DNA damage. Ataxia-telangiectasia (AT), Bloom syndrome (BS), and Fanconi's anemia (FA) are other conditions with DNA repair deficiencies that enhance cancer susceptibility in affected individuals. Mechanisms of tumor promotion and progression. Tumor initiating agents induce mutations, gene rearrangements, or gene amplification and produce a genotypically altered cell. Under the effect of promoting agents which act as mitogens, initiated cells undergo clonal expansion. It is likely that multiple clones of cells are initiated in vivo by an agent that damages DNA. Of these clones, one or a few progresses to malignant cancer through a rare second event. Thus, the promotion phase can be considered as a cell proliferation and clonal expansion stage induced by mutagenic stimuli, whereas the progression phase can be considered as a gradual evolution of phenotypically and genotypically altered cells. This occurs due to genetic instability of the progressing cells and leads to cell heterogeneity within a tumor. In humans, the progression phase may take many years, and during this period, individual tumors develop heterogeneity. They become heterogeneous in their invasive and metastatic characteristics, state of cellular differentiation, antigenic specificity, and responsiveness to drugs, hormones, and immune modulating agents. Apparently, the growth of one progressing cell type is favored over another by some powerful selection processes. The selection process may be due to the development of a growth advantage in the host of a certain cell type over the others. Alternatively, it could be that certain cell types are recognized and destroyed by the host's immunologic defense system better than the others, which provides selection pressure for the expansion of certain clones over the other. Or the process could involve a combination of these factors. In vivo experiments have provided supporting evidence for the existence of such a selection process. Studies on L5178Y lymphoma cell growth in mice have shown that a continual selection of cells with reduced ability to be recognized and killed by cytolytic T lymphocytes (CTL), which are armed against tumors, occurs. The cells are selected until the emergence of a phenotype that is highly resistant to CTL cells. Mechanisms involved in clonal evolution and tumor progression. Malignant tumors arise from a single transformed cell. Various agents such as chemical carcinogens, radiation damage, oncogenic virus, endogenous genetic damage caused by oxidative insult to DNA, or several other potential ways such as hormonal imbalance, chronic infections with H. pylori bacteria, or schistosomiasis parasitic flatworm can bring about transformation. An initiated cell that starts undergoing clonal expansion due to genetic instability acquires multiple genetic changes, which lead to invasive metastatic cancer. Figure 4.67. Figure 4.67 Tumor development stages. Abnormal proliferation of a single mutated cell initiates cancer development. Additional, mutational events and further selection of cells with increased proliferation rate within the tumor cell population results in tumor progression characterized by more rapid growth and malignancy. Studies on colon cancer by Vogelstein and colleagues have indicated that progression occurs in a sequential manner. Colon cancer goes through a series of evolutionary changes starting from hyperplasia, through early-stage adenoma, late-stage adenoma, carcinoma, and finally to metastatic cancer. Weinberg and colleagues, however, have proposed another hypothesis that is supported by clinical data. According to this viewpoint, genes that drive invasiveness and metastasis could be expressed early in the progression pathway. They could actually be the genes that provide a selective growth advantage to these cells. These cells could be present even in early-stage cancers. Hence, it can be said that some cancers almost from the beginning were predestined to evolve into invasive, metastatic tumors. Such a possibility can play a very important role in screening, diagnosis, and choice of therapy for cancers. For example, many women are diagnosed with breast ductal carcinoma in situ based on mammography screening, and likewise, several men are diagnosed with prostate cancer based on testing for a prostate-specific antigen (PSA) followed by a biopsy. Several of these patients have slow-growing tumors such that their overall life expectancy would not be affected, but often they undergo substantial surgical and drug treatments. Therefore, the diagnostic procedures are being evolved such that they are based on molecular profiling, e.g., by gene expression arrays, which will enable distinguishing between the so-called early-stage cancers and help identify those that will be lethal from others. Weinberg theory also states that genetic alterations occurring during tumor progression do not necessarily follow a given sequence and is also suggested that they are different for different cancers. Also, possibly they differ from one patient to another even though they possess cancers of the same histological type. However, ultimately, all these genetic and phenotypic changes result in uncontrolled cell proliferation, and an array of genes need not be identical involved in invasiveness and metastatic ability are expressed. Some of these concepts are supported by clinical data. Studies have shown that gene expression profiling of breast cancer patients is a more efficient predictor of disease outcome than the standard histopathological and clinical staging. This study enabled a more accurate categorization of patients as per prognostic outcome based on actual metastasis-free survival. Additionally, cells with higher tumorigenic potential could be identified based on cell surface markers and isolated from a heterogeneous breast tumor cell population. From these data, it became evident that it is possible to identify aggressive tumorigenic cells in initial tumor biopsies comprising mixed cell populations and can be used as a parameter to distinguish patients possessing potentially more aggressive tumors. Generally, most cancer cases take years to develop an established invasive, metastatic cancer from a small clonal population of initiated cells. The time duration for this process can be 20 years or more. During this time, clonal expansion of an initiated clone of cells takes place via multiple cell doublings. As clonal expansion takes place, several cells in the population acquire multiple genetic alterations. Some of these alterations enable uncontrolled cell proliferation, and some may lead to cell death. The spectrum of genetic alterations acquired can include chromosomal translocations, gene amplifications, gene deletions, point mutations, loss of heterozygosity, LO, and loss of imprinting, LOI. This accumulation of genetic alterations is due to genetic instability. However, the causes underlying genetic instability are not clearly understood, but defects in cell cycle checkpoint controls and defective DNA damage repair are known to be involved. Experimental evidence showing the accumulation of a large number of mutations in cancer cells are available. For example, the study of DNA from colon tumors of patients with hereditary non-polyposis colon cancer (HNPCC) shows alteration in about 100,000 repetitive DNA sequences caused due to defects in DNA mismatch repair in the cells of these patients. Even sporadic cancer cases have been shown to possess mismatch repair defects. A hypothesis called the mutator phenotype hypothesis has been put forth to explain the genetic instability observed in transformed cells, and this has been championed by Loeb and colleagues. The hypothesis states that an initial mutator gene mutation generates further mutations including mutations in additional genetic stability genes, resulting in a cascade of mutations throughout the genome. Molecular defects that could form the basis of this phenotype could be an error-prone DNA replication due to a mutation in DNA polymerases. For this hypothesis to be valid, it is important that the generation of mutator phenotype occurs early in tumorigenesis. Several arguments against this idea include a lack of evidence of an increased mutation rate in cancer cells compared to normal cells, and that clonal expansion of genetically altered cells could give rise to a similar evolution, thus providing cells with a proliferative advantage. Genes and pathways involved in cancer oncogenes. Cell division cycle of somatic cells is regulated by mitogenic signals in order. Binding of growth factors triggers intracellular signaling cascades that permit the progression of cells through the G1S phase of the cell cycle. The complex signaling networks that regulate the entry into the cell cycle are tightly regulated such that a cell divides only when required by the whole organism. It ensures that cells will proliferate during phases such as development, for normal homeostasis, or during an injury. Mutations in the key regulatory genes involved in these signaling networks give rise to cancer. They disrupt the normal regulatory circuits, thereby causing untimely cell division and growth. In addition to disrupted cell division promoting signals, this also includes evasion of signals that trigger cell death, apoptosis, thus defying all normal controls. Although the particular genes that are altered in the sequence in which they are altered may vary from one cancer to another and also from one patient to another, most mutations particularly disrupt a certain number of key regulatory pathways, and hence the genes involved in these pathways. These genes have been identified, characterized, and have been categorized into the oncogenes, tumor suppressors, and caretaker genes. Oncogene discovery and retroviral oncogenes discovery of oncogenes formed a landmark in understanding the molecular and genetic mechanisms involved in cancer. It also contributed in a significant manner to the understanding of the mechanisms underlying the regulation of cell proliferation. In the present times, identifying alterations in the oncogenes has become an integral part of molecular diagnosis in cancer. Also, understanding the mechanisms and pathways involved in oncogenic function have rendered their molecular targets for newer cancer therapies. Although it was known for more than 100 years that viruses could cause cancer, they were first described in 1909 through the study of poultry infecting viruses. The early studies were conducted by Peyton Roos, The early studies were conducted by Peyton Roos, 1911, on the neoplasms induced in the fowls with viral infection. He was the first to suggest that infectious agents could cause cancers. His studies showed that the tumors in the fowl were induced by retroviruses. This idea suggested by Roos was strongly supported by work done by Martin in the 1950s. Martin successfully demonstrated that the tumors induced by the Roos sarcoma virus (RSV) consisted of infected cancer cells. The oncogene in the virus was called as versus SRC. Experimentally, it was also demonstrated by Martin that temperature-sensitive mutants of versus SRC could not transform cells at the non-permissive temperatures. Mike Bishop and coworkers subsequently sequenced the VSRC oncogene, which was identified in the 1970s. Later studies also demonstrated that tumors could be caused by injecting these viruses. The subsequent development of DNA recombinant techniques further advanced the studies in this area. However, a clear understanding of the molecular basis of cancer was achieved only after the cellular homologs of viral cancer-causing components were discovered in 1976. Examination of the viral DNA revealed regions that were very similar to genes that were already present in the animal. This suggested that the viruses at a certain point incorporated sequences of the host genome into their own genomes. By doing so, they also acquired the increased replicative potential for themselves. Around the middle of the 1900s, the concept that cancer cells consisted of mutated cellular genes was well known. But it was work done in the 1970s that advanced this idea to the forefront of cancer research. Work done by Bruce Ames, published in 1975, identified that cancer could be caused by carcinogens, cancer-causing chemicals, through induction of mutations. Alternatively, this also suggested that cells may normally contain genes which, when mutated by some means, may confer a growth advantage and hence cause cancer. Important contributions were made by the studies of Harold Varmus and Michael Bishop. They were awarded the Nobel Prize in Medicine for their contributions in the year 1989. In the year 1976, they identified the cellular homolog CSRC of the viral versus SRC gene. The VSRC was previously described with respect to the chicken sarcoma virus and was hence called as the Roos sarcoma virus (RSV). Earlier studies had suggested that the retroviral oncogenes were derived from normal related genes present in the host cells. Supporting this proposition, normal cells from several species containing retrovirus-related DNA sequences were eventually found. Table 4.10. Further, work done by Varmus and Bishop allowed the unambiguous identification of SRC-related DNA sequences in the normal avian cells through nucleic acid hybridization experiments. The results of these landmark studies altogether identify the presence of DNA sequences closely related to the SRC oncogene in the normal cells and lead to the discovery of the SRC proto-oncogene. Thus, supporting the hypothesis that retroviral oncogenes originated from cellular genes and became incorporated into viral genomes. This further suggested that tumors that were not induced by viruses could have originated due to mutations in related cellular protein cogenes, which directly lead to the discovery of oncogenes associated with human tumors. Consolidating the results of all these studies, it was becoming evident that normal cells contain proto-oncogenes in their DNA, and that under certain circumstances these genes could undergo an alteration and gain the potential to promote cellular growth or transformation. This came to be called as the somatic mutation theory of cancer. Cellular oncogene viral. Oncogene function viral disease. PTGFBB cisplatelet-derived growth factor B subunit. Simian sarcoma. EGFRB ERB epidermal growth factor receptor chicken erythroleukemia. CSF1R versus FMS macrophage colony stimulating factor receptor. McDonough feline sarcoma. HRAS1B ROS cell signaling activation of MAPK cascade. Hardy rat sarcoma. ABL protein tyrosine kinase Abelson mouse leukemia Jun B Jun transcription factor avian sarcoma 17 NYCB transcription factor avian myelosarcomatosis phos transcription factor mouse osteosarcoma table 4.10 cellular and viral oncogenes. The somatic mutation theory was greatly supported by studies performed by Bob Weinberg and colleagues. They showed that when DNA from cancer cells was transferred to the normal NIH3T3 fibroblasts from mice, they could be transformed. When the normal cells were transfected with tumor DNA, they lost the property of contact inhibition and piled up in a discrete area of the culture dish, forming a multi-layered clump of cells. These clumps were called as a focus and displayed contrasting behaviors compared to surrounding normal cells, which cease to grow after forming a sheet of cells of single cell thickness, called a monolayer. It was found that oncogenes transfected from various tumor cells and those of retroviruses such as RSV had similar effects on the behavior of the normal cells. In both cases, normal cellular genes were converted into a transforming oncogene, although the causal agent in one was a retrovirus, and in the other, it was a chemical carcinogen. Further, work carried out on a human bladder cancer gene resulted in the discovery of the ROS oncogene that was homologous to the retroviral oncogene carried by the Harvey sarcoma virus. The viral oncogene was called as the VROS oncogene, and its origin was similar to that of the VSRC oncogene of Rous sarcoma virus. It was concluded that the same normal cellular genes were activated into potent oncogenes either by somatic mutation induced by mutagenic carcinogens or by a retrovirus. The occurrence of the activating point mutation in the ROS oncogene was discovered and was followed by the discovery of mutant forms of other cellular genes such as MYC in human tumors. Presently, a wide variety of mutant genes that are involved in carcinogenesis are known. Further observations indicated tumorigenesis to be a much more complicated process than being caused by mutation in a single gene. It appeared that the development of a tumor extended for a period of several years, and also histopathological analyses of tumor tissue strongly indicated that it was a multi-step process. This implied that human tumors must carry multiple gene mutations and also that a single gene mutation was inadequate to convert a normal cell into a malignant cell. This was proven by laboratory experiments where it was observed that transfection of the normal rat embryo fibroblasts with a clone gene of ROS alone did not cause transformation, but co-transfection with both ROS and MYC oncogenes could transform these cells. Further studies on the cancers of the hematopoietic system led to the identification and characterization of recurrent and clonal cytogenetic abnormalities associated with these cancers. By the cloning of the chromosomal breakpoints involved in translocations and inversions in these cancers, several oncogenes have been identified. More oncogenes were identified by the analysis of the abnormally stained chromosomal regions called the homogeneously staining regions, which indicate gene amplification. Several recurrent karyotypic abnormalities have been observed in many solid tumors and hematologic cancers that include chromosome segments, gain or loss of whole chromosomes, and chromosomal rearrangements. One of the first chromosomal abnormalities identified in human cancers was a small chromosome present in the cells of chronic myelogenous leukemia patients and was called as the Philadelphia chromosome. In the early 1970s, the development of chromosome banding techniques enabled accurate cytogenetic characterization of several chromosomal translocations in human solid tumors, leukemia, and lymphoma. Following the development of molecular cloning techniques, the identification of protein cogenes at the location of the chromosomal breakpoints was enabled in several neoplasms. Proto-oncogenes and oncogenes. Proto-oncogenes are cellular genes that are involved in various cellular functions such as cell division, growth, differentiation, survival, and motility. The products of oncogenes are called as proto-oncoproteins. When these normal genes undergo mutations that activate them permanently and thereby deregulate the functions that are governed by them, they are called as oncogenes, cancer-causing genes. Oncogenes, from Greek onkos, a tumor, generally possess the property of promoting excessive cell division and growth. But they are also shown to play key roles in invasion, loss of differentiation, evasion of apoptosis, and cell motility. Proto-oncogenes are subject to tight regulation in normal cells, thus preventing excessive proliferation, and also inappropriate survival of cells that have accumulated DNA damage. Oncogenes differ from their normal counterpart proto-oncogenes in their hyperactivity and deregulated expression. Oncogenic mutations are dominant mutations, and hence it requires only one copy of the two genes in a cell need undergo a mutation to become overactivated. On the contrary, inactivation of both tumor suppressor gene copies is required for unrestrained cell proliferation and evasion of cell death apoptosis. Such a mutation that requires both copies of the gene to be inactivated are called recessive mutations. Such changes release the normal controls that regulate cell survival or proliferation. However, there are studies that indicate towards the haploinsufficient nature of some tumor suppressor genes. In such conditions, tumorigenesis can be promoted by the inactivation of a single copy of the tumor suppressor gene. Whether oncogenes are activated or tumor suppressor genes are inactivated, they cause similar effects on the cellular behavior. Both result in uncontrolled cell division. Over most of the cellular regulation involve both stimulatory proto-oncogene and inhibitory tumor suppressor components. Oncogenes are involved in the tumorigenesis process as promoters of uncontrolled cell division. Oncogenes include a diverse range of genes that code for proteins involved in various functions such as cell cycle regulation, differentiation, and survival death. As explained in the previous section, oncogenes are the mutated form of the proto-oncogenes and affect cell growth in a dominant fashion by enhancing cellular replication or evading cell death. Nomenclature of oncogenes. Oncogenes are denoted by a three-letter code derived from cancers in which they were identified for the first time. Thus, ROS oncogenes were first described in rat sarcomas. They may carry a prefix distinguishing between cellular C and viral versus homologs, or between differing members mutant forms of the same oncogene family. The CMYC proto-oncogene encodes the transcription factor, CMYC, originally identified as a cellular homolog to the viral oncogene versus MYC of the avian myelocytomatosis retrovirus. There are now several members of the MYC family, CMYC, NMYC, and LMYC, where the prefix denotes the normal cellular C gene, and the dominant tissues in which a particular homolog is expressed, neuronal and lung L. Mechanisms of converting protein cogenes to oncogenes. A genetic alteration in a proto-oncogene converts it into an oncogene. The genetic alteration causes permanent activation, turning on, of the proto-oncogene, converting it into an oncogene. The genetic alterations that result in the formation of an oncogene are called gain-of-function mutations. Gain-of-function mutation. Genetic alterations, mutations that convert a proto-oncogene into an oncogene confer a permanent enhanced activity on the gene product, thus overriding the existing regulatory mechanisms. Therefore, such an enabling genetic alteration is called a gain-of-function mutation. The gain-of-function acts as a dominant allele, i.e., mutation in only one copy of the gene is sufficient to deregulate its function and promote cancer formation. Types of genetic alterations involved in conversion. Various types of genetic alterations, figure 4.68, lead to the conversion of a proto-oncogene to an oncogene and are mostly found either in the coding region resulting in the formation of an abnormal oncoprotein with increased activity or stability, or they may be present in the regulatory elements leading to increased or deregulated protein expression. Figure 4.68, mechanisms involved in the conversion of proto-oncogenes to oncogenes. Proto-oncogene can be converted into an oncogene in several ways. These include point mutations leading to constitutive activation of the protein, gene amplification leading to overproduction of the protein, or chromosomal rearrangements such as translocations that lead to either overexpression or formation of a fusion protein with altered activity. The genetic alterations can range from a point mutation involving a single amino acid of a protein up to major chromosomal rearrangements that entirely deregulate gene expression. Major mechanisms by which oncogenes are activated can be classified as follows. 1. Point mutation. This involves mutations that alter, insert, or delete one to few nucleotides of a gene sequence leading to its permanent activation. The types of mutations that occur include base substitutions, insertions, and deletions. All of these mutations lead to conversion of a proto-oncogene into an oncogene. Mutations in protein cogenes result in structural alterations in their protein products, hence causing their activation. These often lead to a hyperactive gene product with uncontrolled, continuous activity. Also, these alterations can occur in critical protein regulatory regions. For example, base substitutions, insertions, or deletions in the promoter region of a protein cogene can lead to increased transcription. Activating deletions are commonly observed in retroviral oncogenes. Deletions in the amino-terminal ligand binding domain of the retroviral oncogenes such as the ERB, ROS, KIT, MET, and TRK is frequently observed. Most of the characterized oncogene mutations in human tumors are base substitutions that alter a single amino acid within their protein product. Point mutations occur frequently in the ROS family KROS, HROS, and NROS of proto-oncogenes. KROS mutations are predominantly found in carcinomas. Studies have revealed KROS mutations in 90% of pancreatic carcinomas, 50% of colon carcinomas, and 30% of lung adenocarcinomas. NROS mutations are particularly found in hematologic malignancies. Mutations in ROS family are reported in the majority of thyroid carcinomas. Mutation in codon 12 comprises majority of mutations involving the ROS gene. A smaller number of them involve codons 13 or 61. Carcinogen exposure has been associated with ROS mutations in human tumors. Mutations in ROS cause constitutive activation of the ROS protein-mediated signal transduction. Activating point mutations affecting the RET protein cogene involved in MEN2A, multiple endocrine neoplasia type 2A syndrome is another important example. The germline mutation involving cysteine residues of the RET receptor protein is shown to confer ligand-independent activation of its tyrosine kinase activity. 2. Gene amplification. This results in an increase in the number of copies of a gene, thus leading to the overexpression of its protein product. Increase in the copy numbers of a gene within a genome is called as gene amplification. It was first discovered in tumor cell lines. These tumor cell lines acquired resistance to certain growth-inhibiting drugs via gene amplification. Redundant replication of genomic DNA is known to be the cause of gene amplification. This often results in karyotypic abnormalities called as HSRs, homogeneous staining regions, and DMs, double-minute chromosomes. HSRs are chromosomal segments that lack the usual alternating light and dark staining bands. DMs are characteristic minochromosome structures that lack centromeres. Together, HSRs and DMs comprise large regions of amplified genomic DNA consisting of up to several hundred gene copies. Amplification results in increased gene expression, which can in turn confer a selective growth advantage to the cells. Amplification of specific protein cogenes appears to commonly occur in neoplasia, as HSRs and DMs are frequently observed in human tumors. Amplification of three protein cogene families MYC, ROS and ARB, has been shown in significant number of human tumors. CMYC amplification has been shown in about 20% to 30% of ovarian and breast cancers. It has also been frequently reported in certain types of squamous cell carcinomas. NMYC was discovered due to its frequent amplification in neuroblastomas. NMYC amplification is shown to correlate with advanced stages of neuroblastoma, implying a role in tumor progression. Discovery of LMYC was through its amplification in lung small cell carcinoma. Epidermal growth factor receptor ARB amplification has been reported in 10% to 20% of head and neck squamous carcinomas and in up to 50% of glioblastomas. About 15% to 30% of breast and ovarian cancers show ERBB2, HER2, NOI gene amplification. In breast cancer, amplification of this gene correlates with advanced stage and poor prognosis. Amplification of the ROS gene family, including KROS and NROS, has been observed in various carcinomas. Table 4.11 shows some well-known examples of oncogenes activated by amplification in different cancers. Cellular oncogenes, locations, protein functions, types of cancer. ABL9Q34.1 protein tyrosine kinase. Chronic myelogenous leukemia. BCL111Q13.3G1 S specific cyclin D1. Breast cancer, squamous cell carcinoma of the head and neck, bladder cancer. Breast cancer, squamous cell carcinoma of the head and neck, bladder cancer. CDK412Q14 cyclin dependent kinase. Sarcomas. Cellular oncogenes, locations, protein functions, types of cancer. EGFR ERBB17Q12 epidermal growth factor receptor. Glioblastoma multiforme, epidermoid carcinoma, bladder. Cancer, breast cancer. ERBB2, NOI17Q12 Q21 growth factor receptor. Breast cancer. Ovarian cancer. Stomach cancer, renal adenocarcinoma of salivary gland, colon carcinoma. MDM212 Q14.3-Q15 P53 binding protein. Sarcomas. MET7Q31 hepatocyte growth factor receptor. Amplified in cell lines from human tumors of non-hematopoietic origin, particularly gastric tumors. MYB6Q22 Q23 DNA binding protein essential. For normal. Hematopoiesis. Leukemias, colon carcinoma, melanoma. MYC. CMYC 824.12 Q24.13 DNA binding protein. Small cell lung cancer, giant cell carcinoma of lung, breast cancer, colon carcinoma, acute promyelocytic leukemia, cervical cancer, gastric adenocarcinoma, chronic granulocytic leukemia. MYC and NMYC 2P24.3 DNA binding protein. Neuroblastoma, small cell lung cancer, retinoblastoma, medulloblastoma, glioblastoma, rhabdomyosarcoma. Adenocarcinoma of lung, astrocytoma. MYCL1, LMYC, 1P32 7P15 serine, threonine protein kinase, small cell lung cancer. Cellular oncogenes, locations, protein functions, types of cancer, MYCLK1. RAF13P25 GTPase, non-small cell lung cancer. HRAS111P15.5 GTPase, bladder cancer. CRAS212P12.1 GTPase, adrenocortical tumor, giant. Cell carcinoma of lung, NRAS1P13 GTPase, breast cancer. Table 4.11 list of oncogenes activated by amplification in cancer. 3. Chromosomal rearrangements. Recurrent chromosomal rearrangements are frequently associated with hematologic malignancies and some solid tumors. Usually, the arrangement involves a chromosomal translocation. However, chromosomal inversions can also occur occasionally. A chromosomal translocation, resulting in the relocation of a proto-oncogene into a new chromosomal site, can lead to its transcriptional activation, leading to higher or inappropriate gene expression, or can result in a fusion of the proto-oncogene with another gene, thus producing a fusion protein with an altered function, making it oncogenic. A chromosomal translocation leading to the formation of a gene activation. The translocation T8, 14Q24, Q32 between chromosome 8 and 14 is found in about 85% of Burkitt lymphoma cases. Figure 4.69 CMYC translocation. Translocation of CMYC proto-oncogene from chromosome 8 to chromosome 14 IGH, immunoglobulin heavy chain, locus in Burkitt's lymphoma, leading to abnormal CMYC expression. This is a well-characterized example involving transcriptional activation of a proto-oncogene. As a result of this translocation, the CMYC gene, originally located at chromosome band 8Q24, is placed under the control of immunoglobulin heavy chain regulatory elements, located at chromosome band 14Q32. This results in the activation of CMYC-encoded nuclear protein that regulates cell proliferation, leading to the development of Burkitt lymphoma. Some cases of Burkitt lymphoma also show translocations involving immunoglobulin light chain genes. However, the translocations in individual cases of Burkitt lymphoma may vary significantly with respect to the chromosomal breakpoints relative to CMYC gene. The consequence remains the same. All lead to deregulation of CMYC expression, resulting in uncontrolled cellular proliferation. The CMYC gene activation involving translocation T8, 14Q24, Q11 has been observed in some T-cell acute lymphoblastic leukemia (TL) cases. In these cases, CMYC transcription is placed under T-cell receptor alpha regulatory elements located at 14Q11. In addition to CMYC, translocations involving several other oncogenes has also been reported for TL. The proteins encoded by these genes include several transcription factors that function through DNA binding and protein-protein interactions. Inappropriate expression or overexpression of these proteins in T-cells is suggested to inhibit T-cell differentiation and cause uncontrolled cellular proliferation. Activation of several other protein cogenes via chromosomal translocations has been reported in leukemia and lymphoma cases. Activation of BCL2 gene located at 18Q21 has been observed in some large cell lymphomas and most follicular lymphomas. The BCL2 activation occurred due to AT14, 18Q32, Q21 translocation. The overexpressed BCL2 protein inhibits apoptosis, causing an imbalance between lymphocyte proliferation and programmed cell death. Activation of cyclin D1 BCL1 gene located at 11Q13 due to AT11, 14Q13, Q32 translocation has been shown in mantle cell lymphomas. The cyclin D1 gene encodes cyclin D1, AG1 cyclin involved in normal cell cycle regulation. B chromosomal translocation leading to the formation of a gene fusion. Several abnormal chromosomes associated with cancers have been identified by examining chromosomes through karyotyping procedures using tumor cells. Chromosomal translocations are one of the most frequently observed chromosomal aberrations. The translocation T9, 22Q34, Q11 involving chromosomes 9 and 22 were observed in the leukemic cells of most chronic myelogenous leukemia (CML) patients. This translocation fuses the CABL gene, normally located at 9Q34, with the BCR gene at 22Q11. Figure 4.70 gene fusion due to chromosomal translocation. In CML, chronic myelogenous leukemia, a oncogenic chimeric protein of 210 kDa is produced due to the fusion of the CADL gene with the BCR gene, occurred due to T922q34, q11 translocation. The BCR-ABL fusion chimeric protein, 210 kDa, an outcome of this translocation event, created on the derivative 22 order 22 chromosome, consisted of an amino terminus of the BCR joined with carboxyl terminus of ABL, a protein tyrosine kinase. The fusion protein is characterized by increased tyrosine kinase activity and abnormal cellular localization. This resulted in inappropriate activation of the ABL protein tyrosine kinase activity in hematopoietic cells, causing uncontrolled proliferation by activating several pathways which are otherwise regulated by extrinsic growth factors. This translocation event generates two abnormal chromosomes. It resulted in a longer chromosome 9 and a smaller chromosome 22. This smaller chromosome was named Philadelphia chromosome because it was first recorded in the city of Philadelphia. About 20% of acute lymphoblastic leukemia, all cases also consist of the T922 translocation. But in these cases, the BCR gene breakpoint differs from that found in CML, and a BCR-ABL fusion protein of 185 kDa is formed. Several proto-oncogenes such as CMYC, EWS, and RET have been shown to be involved in several translocation events. Figure 4.71 chromosomal translocation in CML. Translocation occurs between the chromosome 9 and chromosome 22, leading to the fusion of CADL gene and BCR gene. The translocation event results in a longer chromosome 9 and a smaller chromosome 22 called the Philadelphia chromosome. Types of oncogenes. Majority of the oncogenes function in pathways that usually regulate normal cell division and cell survival. On the basis of functions and biochemical properties of the proteins encoded by the proto-oncogenes, the corresponding oncogenes can be divided into 5 groups which include 1. Growth factors. 2. Growth factor receptors. 3. Signal transducers. 4. Transcription factors. And 5. Regulators of cell death and others. Oncogene chromosome discovery cancer means of activation protein. Growth factors. B622q12.3-13.1 viral homolog glioma fibrosarcoma. Constitutive synthesis. B-chain PDGF. Growth factor tyrosine kinases. HER1 EGFR 7p1.11.3 DNA amplification. Squamous cell carcinoma. Gene. Amplification increased. Protein. EGF receptor. Oncogene chromosome discovery cancer means of activation protein. HER2 Noi17q11.212 mutation. Amplification. Neuroblastoma breast. Carcinoma gene. Amplification human EGFR2 preferred. Heterodimerization partner for other EGFR family members. HER3 amplification squamous carcinoma. Gene. Amplification. Signal transduction pathways. Non-receptor tyrosine kinases. ABL9q34.1 chromosome CML DNA rearrangement. Translocation. Fusion. Proteins. Tyrosine kinase. BCR-ABL chromosome CML fusion protein tyrosine kinase. SRC20p12-13 viral homolog colon carcinoma. Constitutive activation tyrosine kinase. Membrane-associated G proteins. HROS11p15.5 viral homolog transfection. Colon, lung, pancreas carcinomas point. Mutation GT case. ROS12p11.1-12.1 viral homolog transfection. AML, thyroid, carcinoma, melanoma point. Mutation GT case. NROS1p11-13 transfection carcinoma, melanoma. Point. Mutation GT case. Serine threonine kinases, cytoplasmic. MOSH-Q11, MOS, viral homolog sarcoma constitutive activation. Protein kinase SRTHR. RAF3P25, RAF1, viral homolog sarcoma constitutive activation. Protein kinase SRTHR. PIM16P21, PIM1, insertional mutagenesis. T cell. Lymphoma constitutive activation protein kinase SRTHR. Oncogene chromosome discovery cancer means of activation protein. Cytoplasmic regulators. CREEK17P13, CREEK viral homolog breast constitutive tyrosine. Kinase activity. SH2-SH3 adapter. PRAD1 cyclin D1. Transcription factors. Phos14q21-22 viral homolog. Osteosarcoma deregulated activity. Transcription factor P. JUN-P31-32 viral homolog. Sarcoma deregulated activity. Transcription factor P. CMYC8q24.1 MYC viral homolog. Multiple. Cancers, breast, stomach, lung, cervix, colon, neuroblastomas. Burkitt's. Lymphoma amplification. Lymphoma amplification, translocation deregulation transcription factor. NMYC2P24 DNA amplification. Neuroblastoma, lung carcinoma deregulatory activity. Transcription factor. LMYC1P32 DNA amplification. Carcinoma of lung. Deregulatory activity. Transcription factor. MYB6Q22-24 viral homolog myeloblastosis deregulated activity. Transcription factor. Apoptosis regulators and others. BCL218q31.3 chromosomal translocation. B cell. Lymphomas constitutive activity. Anti-apoptotic protein. MDM212q14 DNA amplification. Sarcomas gene. Amplification. Increased. Protein. Complexes with P53. AML equals acute myeloid leukemia, CML, chronic myelogenous leukemia, CSF equals colony stimulating factor, EGF equals epidermal growth factor, FGF equals fibroblast growth factor, HGF equals hepatocyte growth factor, NGF equals nerve growth factor, PDGF equals platelet derived growth factor. A diverse range of genes comprises the oncogenes. These include proteins involved in cell cycle control, differentiation and cell death survival. They can be categorized into 5 functional classes, including growth factors, growth factor receptors, signal transducers, transcription factors, and survival proteins. The figure represents the key examples in each of the categories. 1. Growth factors invariably oncogenic activation of growth factors GF is caused due to transcriptional activation of the gene, resulting in growth factor overproduction. CIS, which is one of the best known GF oncogenes, is incorporated into the simian sarcoma virus genome versus CIS, and it encodes PDGFB platelet-derived growth factor chain. The mutated gene results in platelet-derived growth factor (PDGF) overproduction. Increased PDGF has been observed in several cancers, such as fibrosarcomas, tumors of fibrous connective tissue, and gliomas. 2. Growth factor receptors of receptor tyrosine kinases. Several mutations result in the constitutive activation of receptor tyrosine kinases (RTKs). B. HER2 Noi. The HER2 Noi, also called ERBB2 gene encodes the human epidermal. Growth factor receptor type 2, normal. Cells express this receptor at moderate levels. The gene is involved in cellular responses to growth factors, i.e., on binding of a growth factor, it triggers cell division. About 20% of human breast cancers show amplification of the HER2 NOI gene, which in turn leads to an overexpression of the HER2 cell surface receptors. The breast cancer cells overexpressing HER2 receptors show about 40 to 100 times higher expression as compared to normal cells. The overexpression of the receptor leads to uncontrolled cell proliferation, both tumor progression and response to treatment are also known to be altered due to HER2 NOI gene amplification. Overexpression of HER2 NOI is suggested to be attributed to tumors with more aggressive behavior. Also, it is known to make tumors more sensitive to chemotherapy. Breast cancers characterized by the HER2 NOI gene amplification are referred to as HER2-positive breast cancer. CEGFR. Another example of a receptor tyrosine kinase altered in cancers is the epidermal growth factor receptor. EGFR. Alteration of the same has been reported in about 15% of Figure 4.72 representatives of the different groups of oncogenes. Small cell lung cancers. Alteration in the EGFR gene has been observed in several other cancers such as breast, esophagus, head, and neck, and so on. Inappropriate activation of EGFR is known to occur mostly due to a point mutation or a gene amplification. Transcriptional upregulation has also been reported. Increased activity of the EGFR leads to excessive cellular proliferation, increased cell survival, angiogenesis, and metastasis. DPDGFR. The platelet-derived growth factor PDGF receptor PDGFR is converted to an oncogene in some human leukemias. The oncogenic conversion occurs due to a chromosome translocation as a result of which the amino terminus of the PDGF receptor is replaced by amino-terminal sequences of a transcription factor called telephone. The fusion protein telephone PDGFR that is formed is capable of dimerization even without growth factor binding, hence resulting in constitutive activation of the intracellular kinase domain. This in turn results in unregulated proliferative signaling from the oncogenic protein, e.g. protein-coupled receptors. The MOS gene is one of the well-known examples of oncogenic G protein-coupled receptors. It was first identified in tumors of mammary carcinoma. The involvement of the MOS gene in human epidermoid tumors has also been suggested. This gene has been shown to encode the angiotensin receptor. Another example of a G protein-coupled receptor is the thyrotropin receptor TSH receptor. Mutations in the thyrotropin receptor gene converted into an oncogene found in thyroid tumors. The thyrotropin hormone, pituitary hormone, is involved in stimulation of thyroid cell proliferation, and it functions through the TSH receptor. Mutations of the thyrotropin receptor result in their constitutive activity, which drives cell proliferation through CAMP signaling pathway activation. 3. Signal transducers of non-receptor membrane-associated tyrosine kinases. The SRC protein is a tyrosine kinase, the BSRC gene being the first oncogene to be discovered is of historical significance. Besides, CSRC, the normal cellular gene is a typical protein tyrosine kinase. The functional domains of the tyrosine kinases that were subsequently discovered were referred to as SH domains, where SH stands for SRC homology, indicating homology to those in SRC. An overexpression of SRC has been shown in several human cancers such as rhabdomyosarcoma, neuroblastoma, and carcinomas of colon, small cell lung, and breast. Beat membrane-associated G proteins, the ROS family. The ROS genes are frequently mutated in cancers and are amongst the first human oncogenes to be identified. The ROS family genes encode the membrane-associated small G proteins. 3 different homologs of the CROS oncogene have been identified, and each of them was found in different types of tumor cells. In all cases, point mutations were observed to be the cause of oncogenic conversion of ROS genes, and the mutation resulted in the loss of intrinsic GTPase activity of the normal ROS protein. As a result of this, the ROS becomes permanently activated, switched on. In addition to gene mutations, overexpression and amplification of ROS gene can also lead to development of cancer. The altered ROS can affect signaling pathways involved in regulation of cell division and cell death, thereby resulting in uncontrolled proliferation of cells. ROS mutations have been reported in about 90% of pancreatic cancers, 50% of colon and thyroid cancers, and about 30% of lung cancers. It has also been reported in several other cancers such as bladder, breast, ovarian, kidney, skin, liver, and some leukemias. C. Membrane-associated large G proteins in some cell types. The genes encoding G proteins also act as oncogenes. The GSP oncogene, which encodes GS alpha subunit, is a result of point mutations similar to those found in ROS. These mutations cause constitutive activation of the GS alpha subunit, which in turn leads to unregulated adenylyl cyclase stimulation. The GSP oncogene has been shown to be involved in pituitary and thyroid tumors. Conversion of other G protein alpha subunits to oncogenes via similar mutations has been reported in other cell types, including ovarian and adrenal tumors. Deep serine threonine kinases. The RAF fibrosarcoma gene was found in association RAF fibrosarcoma virus as a transforming oncogene. Incorporation of the RAF gene into the viral genome resulted in truncated protein which acquired constitutive kinase activity. As the function of the normal RAF gene product, RAF is to phosphorylate threonine of the MAP kinase MAPK following receptor activation. The oncogenic RAF results in constitutive activation of the downstream MAPK pathway. The mutation of the RAF gene has been reported in some human cancers. 4. Transcription factors. Oncogenic activity has been reported for a substantial number of deregulated transcription factors. It results in an altered expression of several genes. Some of the well-known oncogenic transcription factors are FOS, JUN, MYC, NFKB, GLI, MYB, ETS, etc. MYC. MYC protein is a transcription factor regulating the expression of several genes and plays a key role in several well-known human cancers. It is an oncogene initially identified in the avian myelofibromatous virus, CMYC. MYC gene mutations have been reported in many different cancers, including Burkitt's lymphoma, B-cell leukemia, and lung cancer. The MYC genes may also become activated by amplification or gene rearrangement. Gene rearrangements occur due to chromosomal translocations, e.g., translocation between chromosomes 8 and 14, see section D5.2.1.4.2, has been shown to result in MYC overexpression, leading to development of B-cell lymphoma. In addition, activation of this oncogene is known to occur at some stage of tumorigenesis in majority of human cancers. Although the mechanism of CMYC activation in several cancers is unclear, there is evidence of mutations conferring increased stability to the CMYC protein. Invariably, in all cases, deregulated or increased level CMYC expression is known to cause the transformation. B. FOS. FOS gene activation is caused due to truncations and consequent loss of coding and non-coding gene sequences following incorporation into genome of the virus. Such changes lead to the loss of sequences involved in transcriptional repression of FOS and also the loss of disability sequences. Consequently, a stable FOS protein is formed. Contrary to the normal condition in which FOS is expressed transiently in replicating cells, the altered FOS may persist at high levels throughout the cell cycle. See ETS. ETS family. ETS transcription factors, which are the nuclear targets downstream of the ROS MAP kinase signaling pathway, are characterized by a conserved ETS domain. Deregulation of the ETS genes causes malignant transformation of cells. Rearrangement of several ETS genes to produce chimeric oncoproteins has been observed in human tumors and in human leukemia. These transcription factors are involved in processes such cell can undergo. When the cell reaches the limit of telomere shortening, it enters cellular senescence or growth arrest. Cancer cells can continue to replicate without reaching the state of senescence. Telomerase is the enzyme involved in maintenance of chromosome ends. Telomerase is expressed in cells during fetal development, while most adult human cells do not express telomerase. However, in certain cancer cells, reactivation of the telomerase enabling unlimited replication has been observed. Hence, the gene encoding the active telomerase enzyme component, TERT, is considered as a protein because unregulated cellular proliferation is caused due to its abnormal expression. 3. Tumor suppressor genes. A tumor suppressor is a protein that slows or inhibits cell proliferation and prevents damage or abnormal cells from becoming malignant if they have been exposed to DNA-damaging agents, such as UV or X-rays, or to hyperproliferative signals induced by oncogenes. Induced expression of tumor suppressor genes in normal cells brings about growth arrest or apoptosis, based on the type of cell and the level of expression. On the contrary, functional loss of tumor suppressor makes the cell susceptible to tumor formation by providing a proliferative advantage. This in turn usually causes accumulation of genetic alterations, ultimately leading to formation of aggressive cancers. Tumor suppressors are guardians of the cells against DNA damage. They protect the cells against DNA damage induced by ultraviolet, UV, exposure to sunlight, gamma radiation, X-rays, or chemotherapeutic drugs. They guard the cells against excessive abnormal proliferative signals. Proliferation of such cells is arrested, and they are induced to commit suicide, apoptotic cell death, and thus are prevented from becoming malignant. Tumor suppressors oversee critical cellular checkpoints involved in governance of meiotic cell cycle, as invasion, metastasis, and neoangiogenesis, thus playing a significant role in malignant transformation and tumor progression. 5. Regulators of cell survival and death. BCL2, B-cell lymphoma gene 2, family members are the protein cogenes specifically involved in regulation of programmed cell death. These were first identified through the studies on chromosomal translocations that occur in human lymphoma. The product of BCL2 gene, BCL2 protein is localized to the endoplasmic reticulum, nuclear membrane, and inner mitochondrial membrane. BCL2 family includes a large number of either pro or anti-apoptotic proteins. If there is an increase in expression of anti-apoptotic BCL2 family members or loss of pro-apoptotic members, the cells become susceptible to other cancer-inducing mutations, particularly those which promote cell proliferation. These cells lose their tumor suppressive ability and escape normal cell death mechanisms, thus surviving to give rise to cancer. Other oncogenes. 1. Cyclin D growth factor stimulation activates intracellular signaling pathways, which ultimately regulate cell cycle machinery components that promote progression through G1 restriction point. Growth factor stimulation induces the D-type cyclins, and these play an important role in coupling growth factor signaling to cell cycle progression. The cyclin D1 gene is a proto-oncogene, which is activated into an oncogene called D1 by gene amplification or chromosome translocation. These alterations cause constitutive cyclin D1 expression driving cell proliferation even in the absence of stimulation by growth factor. 2. Telomerase. The DNA replication process causes the shortening of chromosome ends telomeres during each cell division, and hence limits the number of cell division cycles of given differentiation, apoptosis, DNA repair, and transcription. Certain tumor suppressors inhibit the inappropriate activation of cell growth signaling pathways. If the tumor suppressors undergo a mutation, deletion, or gene silencing, they become functionally inactivated. This results in an imbalance between cell proliferation, differentiation, and cell death, which facilitates tumorigenesis. Rarely, certain tumor suppressor gene mutations are inherited by individuals that predispose them to cancer. Presently, tumor suppressors represent about 0.001% of the total number of genes that comprise the entire mammalian genome. With the arrival of novel technologies to study cancer cells, this number may increase. It can be said that the evolution of tumor suppressors is an integral part of multiple elaborate defense systems formed to prevent abnormal cell proliferation. Usually, proteins that inhibit cell proliferation in one way or another are encoded by tumor suppressor genes. Loss of one or more of these is known to be involved in the development of several cancers. Generally, 5 broad classes of proteins encoded by tumor suppressor genes have been identified. Y intracellular proteins, e.g., Rb, P16 cyclin kinase inhibitor that regulate or inhibit cell cycle progression. Y hormone receptors, e.g., tumor-derived growth factor beta involved in inhibition of cell proliferation. Y checkpoint control proteins involved in cell cycle arrest in response to DNA damage or abnormal chromosomes, e.g., ATM, P53. Y apoptosis promoting proteins, e.g., P53. TGF beta. Y DNA repair enzymes, MSH2, MLH1. DNA repair enzymes, etc. are not directly involved in inhibition of cell proliferation. However, cells that lose DNA repair ability tend to accumulate mutations in several genes, which include the critical cell growth and proliferation controlling genes. Thus, loss-of-function mutations in DNA repair enzyme genes lead to activation of oncogenes and inactivation of tumor suppressor genes. 1. Classification of tumor suppressor genes. Tumor suppressor genes exhibit a broad range of biochemical functions. They function in a wide range of regulatory networks, including DNA damage response and cell cycle regulation. Tumor suppressor genes can be categorized into two main groups: caretakers and gatekeepers.